Welcome to our course, Bioimage Analysis. In this video, video 3, we're looking at EBImage for image analysis. So what we want to achieve is to read in and write out images in R. We will manipulate images in R and also filter and transform them and perform things like segmentation and thresholding to extract particular features that are of interest, and also look at cells' spatial distributions. So in terms of viewing image types, we can look at PNG, TIFF, JPEG, standard image formats. We can also look at cell assay data so that we can segment cells and extract quantitative information from that. And we can look at an example of segmentation on the right where we identify different cellular features that are of interest. So what we're trying to do here is resolve cell structures, the proteins, morphology changes, virus particles, antibodies, and so on. And we might ask, are there cells there? Where are they? And what's the proximity of other things like virus particles to them? Can we identify different membranes and nuclei? And what we're thinking about with our image is that our image is a set of pixels, albeit with varied resolutions. And those pixels have quantitative information on that, typically 3 channels, red, green, blue. And therefore, we're really thinking about how we take in our PNG and take our image X and how we can transform it. So we have different pieces of information inside that image, and we can adjust them by modifying intensities and so on, and rescaling them background and things like that. So we can think where we have brightness and things, B is maybe the contrast. So if we look at image like this on the right, what essentially is recording is levels of brightness here going from the middle where we have our object to the outside where it's much darker. And we can scale that, we can modify that scale. So if we import an image, we can do all sorts of different things to alter it in the way that suits us. So for example, we might want to look at 2 different RNA interference reagents on human HeLa cells using fluorescence microscopy. I might have a negative control as for one collection of cells, and then also ones that are treated with treatment of interest, and that's taken from this study. So you might want to think about how do we get phenotypes from this image, where we've got 3 different layers of actin, tubulin, and DNA different together that we can merge into one image. So we've got the same 3 channels containing the same population of HeLa cells that are perturbed by this Si or Luc element. We can combine these channels and then create our image on the right containing those 3 different channels. And we'll look at how we can assess these different images, the 3 different images separately, and then also the merged image as well. Once we have this image B, we can do different things. One common task then is to do segmentation, where we're splitting an image into multiple objects. So you might say, okay, this is clearly a parrot here, and this one is a tiger. And therefore we can use different tools to identify what is present given our background. And that gets more complex when we have low resolution images or ones that are not very bright. There's different types of segmentation as well. So we can have simply semantic segmentation where it's simply binary, black and white, for example. Instant segmentation has integers for different colors depending on what features we have present. So slightly different approaches. We can also think about different masks. So for binary masks, the pixels are either 1s or 0s, like our example previously. On the right, we can have an integer mask where they have a different value, and a boundary mask, such as one over here, where the pixels are binary except the boundaries. So for EBImage, what we can do is look at things like staining different nuclei. So we use some sort of thresholding to identify them, and morphological opening connected set labeling to identify where the nuclei are. We can stain cell membranes from using Voronoi segmentation, using the nuclei as seeds. And then also following that, then estimate the cell shapes. So we can look at the cell sizes. So they might have particular volume, where there might be some sort of difference that's of interest to our experiment. And we can look at the distribution of cell shapes and their frequency on the y-axis and the sizes here on the y-axis. So you can see here there's a clear difference between our treatment and control. There's a definitely the treatment ones are larger. We can also consider there we may have blurry images, and therefore we can think about how to optimize this images to interpret the shapes more clearly. So we might have an original one like this. And if we do thresholding on that, we'll get poor pixelation resolution. Whereas if we average that, so essentially each pixel borrows the average pixel intensity from the ones around it, it can make the thresholding process and quantification of areas of interest more easy. We can also think about that we're dealing with pixel intensity arrays. So for each, if we had a grayscale image like that, each pixel contains a value of brightness. And then if we have multiple channels, so we could have things like time at the XYZ for blue, green, red, together three different channels. We have different things that we can consider XYZ dimensions as well. And therefore, if we're trying to bring up a single image, we can interpret that in one way as different channels, as the merged image, and then also images changing over time too. So that's what we'll explore in this course, is how we can kind of create different images so you can see the same image with different levels of brightness here, and then also warp them like the RX image on the right here.